There are a couple of words I want you to understand before we start with Chapter 9. The first one is if we say muscle fiber, we're really talking about a skeletal muscle cell. They're the same thing. This is an electromicrograph of a muscle fiber. The whole big picture is the muscle itself, and it's divided into individual muscle cells or fibers. If you put a roast into a slow cooker, when you put the roast in there, it's one big, large hunk of meat. But there are membranes that actually separate individual muscle fibers or cells and individual bundles of cells. So as the roast cooks, the individual fibers separate from each other, and that's why a very tender roast looks real stringy, because those are the muscle fibers. Something else I'd like you to know is that the prefix myo, mesi, and sarco all mean muscles. So we're going to be talking about the sarcolemma, which is the plasma membrane around a muscle cell. We're going to be talking about myofibrils and myofilaments. Those are structures of the muscle. And then mesosomes or myceliums are going to be membranes around the muscle. So let's begin Chapter 9. This first screen is actually a review of Chapter 4 when we talked about the 4 tissue types. If you remember, the 4 tissue types are epithelium, connective, muscle, and nervous. And when we focused on muscle, we said there were 3 types of muscle: skeletal muscle, cardiac muscle, and smooth muscle. Do you remember some of the characteristics about skeletal muscle? This is a picture of what skeletal muscle looks like under the microscope, an artist's drawing of skeletal muscle cell, and then an example in the body. We know that skeletal muscles mostly move bone. Those muscles that are attached to skin in the face do give us facial expressions, but most move bone. If you look at the microscope image, you can see that there are stripes or striations, and this is the arrangement of the proteins in the muscle. And then we know that we can control our skeletal muscles, so they're voluntary. Here's a picture of cardiac muscle. How does it compare to skeletal muscle? Well, we still have the striations, but do we control our heart? Can we say heart beat faster or slower? No, so it's involuntary, and of course cardiac deals with the heart. The last type of muscle is smooth muscle, and to me, it really doesn't look smooth at all. It's the individual smooth muscle cells are fat in the middle and tapered at each end, and so they're not striated. The muscle proteins are not overlapping rows like they are in cardiac and skeletal. So it's smooth. It is involuntary like cardiac muscles, and where we find smooth muscle is in hollow organs. So it moves substances through hollow organs. That's the smooth muscle. So that should be a review. What we're going to focus on mostly as we talk in Chapter 9, we're mostly going to talk about skeletal muscle. Towards the end, we'll talk a little bit more about smooth and cardiac again, but mostly focusing on skeletal muscles. Now, when you look at muscles, they have 3 functions. One function is that it produces movement. If we're moving an entire body, we might say that that movement is locomotion. Or if we're using a body part to manipulate the environment or change the environment, that type of movement might be manipulation. Or if when our muscle contracts, it appears to get smaller because it's been hit by a nerve impulse, that would be a response. Or if our small intestine were to have peristaltic waves that propelled food through it, propulsion would be a type of movement as well. Another function that muscle has is that it maintains posture. It enables us to keep our skeletal system extended and to keep perpendicular to our floor, our surface. Also, it helps us to stabilize our joints in that extended position. If it weren't for the muscles, we'd just be a heap of bones and tissue. We really couldn't use our bones effectively. And the last function of muscle is thermogenesis, creating temperature. Our muscles are considered machines. They're actually part of our lever systems in our body. But when a machine does an action or works, it has to have energy. And there's no machine that's 100% efficient. And so one of the waste products that's given off when we don't use all of our ATP efficiently is heat. But in the human body, and like other mammals as well, we use that heat to maintain body temperature so our enzymes and chemical reactions can occur. When we look at the muscle and how it functions, we can see 4 characteristics of muscle that enable it to do the functions we just mentioned. One of those is the ability to respond to a stimulus. We looked at that in Chapter 1 because we know all living things have the ability to respond to a stimulus. Sometimes we call that responsiveness, sometimes excitability, other times irritability. Now, when a muscle is excited or responds, it does that. What it does, if ATP and calcium are present, is that it shortens. Nothing in the muscle really shortens, but products or proteins in the muscle end up sliding across each other or contracting. So contractility, the ability to shorten, is the second functional characteristic of muscle. Now, when a muscle on the front side of your limb contracts, the muscle on the back side of the limb has to be able to stretch or extend beyond its resting position sometime. That characteristic is called extensibility. And then when the contraction is over, the muscle and the other units of the muscle, like the tendons, have to have the ability to go back to their original length, and that is called elasticity. So for a muscle to be able to work and function efficiently, it must have excitability, contractility, extensibility, and elasticity. So now let's move to the gross anatomy of a skeletal muscle. When we look at a muscle, when we look at the biceps brachii or the biceps femoris, we're looking at a distinct organ, that is a muscle. We can take that big muscle group and divide it into smaller parts. Your body does that over and over as it increases its surface area to work more efficiently. If we were to cut open a big muscle and look into it, we would see that the muscle cells or the fibers are arranged in bundles, and those bundles are called fascicles. So what is a fascicle? A fascicle is a bundle of muscle fibers or cells, and you can see in this picture that these are each bundles or fascicles, and inside the bundle or fascicles are muscle fibers. So muscle fibers, we know, are called muscle cells, and if we were to look inside of a muscle cell, we would see some structures called myofibrils. Protein structures called myofibrils. They extend the whole length of the cell. If we were to break myofibrils down into smaller components, we could see that the myofibril is made of thick proteins and thin proteins. Those thick and thin proteins are called myofilaments. The thin myofilament is actin. Actin is thin, and the thick one is myosin. Now, there are other myofilaments in the muscle, and we'll talk about those later, but these are the two main ones. So we've gone from a muscle unit to fascicles to fibers to myofibrils to myofilaments, actin and myosin. And here's an enlarged view of the myosin being the thick and the actin being the thin blue one. So how do we hold our muscle cell in this unique arrangement of fascicles, fibers? We do that with membranes. There are 3 basic membranes that help us organize our muscle structure. The one that's on the very outside of the whole muscle unit is called the epimysium. Some people say epimysium. I've heard it said both ways, but the epimysium surrounds the whole muscle unit. The membrane that surrounds each fascicle is called the perimysium, and then separating the muscle fibers is the endomysium. So from superficial to deep, we have epimysium, perimysium, endomysium. And we say they're individual membranes, but actually it's one great big huge membrane that would wrap around the muscle and then invaginate to wrap around the bundles and then invaginate to wrap around the outside of the muscle fibers. And this is what I was talking about earlier, that if you cook a roast, you are degrading or melting away those membranes, and so all those muscular structures just get unorganized and fall away from each other. Now these membranes also sometime will extend past the body or the main part of the muscle and become what we know as tendons. So let's practice a little bit. I'll hesitate for a minute and see if you can name 1, 2, 3, and 4, which is organization of muscular structures, and then 5, 6, 7, which are the membranes that give us that organization. So 1 is the muscle unit, 2 is the fascicle, 3 is the fiber, and 4 is the myofibril. Around the whole muscle unit is the epimysium, around the fascicle is the perimysium, and around the fibers is the endomysium. So you're probably wondering, that's a lot of information. How am I going to study that, Miss Anderson? Well, you can go to the study area in mastering, and there are actual activities that you can do. You can see that same picture. This is where I got it from. You can practice labeling those structures. Also, if you go to the quizzes on the study area, there are questions, multiple choice questions there that I use a lot on test, hint, hint, that would be good study areas too. For example, number 1, the entire skeletal muscle is surrounded by, which one is that? It would be the epimysium. And what's nice about these quizzes, and if you'll see over there in the left-hand corner, games and activities and actually videos, is that you can watch these as many times as you would like and then go and do your homework. And they're very similar to the homework. When you do the homework, you get the grade. In some of those movies that are to the side, some of them are IPs or interactive physiologies, and you'll notice this one topic right here, anatomy review, the skeletal muscle tissue. They do just what I just did in the lecture. They go from the big muscle to the fascicles to the fibers to the myofibrils, and they even go microscopic. And notice while we're here that there are some topics, the neuromuscular junction, muscle metabolism, sliding filament theory. All of these are topics that we'll discuss, and this is an excellent place to go and to listen to somebody else's point of view, get a little twist on it, and maybe come to a better understanding. There are also video tutors and another kind of video over these same topics. So there's lots of resources on your study area in the MyLabs Plus. So let's continue with the note. For each one of our major muscles, each muscle is going to be served by one nerve and one artery and more than one vein. And those vessels or that nerve is actually going to enter into the thick part of the muscle, which we call the belly or the body part. Once it goes in, it's going to start branching profusely, and it's going to go up through those membranes so that it can innervate or it can supply or drain groups of muscle cells. Now, when we look at muscles, they need lots of oxygen and nutrients. We have to break glucose down using oxygen aerobically to produce ATP. Now, you're going to see later in the notes there are other ways to get ATP quicker, but this is the most efficient way. But if we use oxygen to break down glucose, we're going to get rid of or give off carbon dioxide, so we need a way to get rid of carbon dioxide. So that's why muscles are very vascular. Now, how do muscles attach to bones and to cartilage and other structures? Most muscles will span a joint and attach to the bone in at least 2 places. And the end of the muscle that attaches to the movable bone, this is something we talked about in Chapter 10, the end that attaches to the movable bone, so in this case, this would be the movable bone. This end, do you remember what it was called? Insertion, and the end that's attached to the more stationary or the less movable bone is the origin. Remember in Chapter 10 you were given several muscles you have to know the origin and insertion for. So looking at this picture, if this is the insertion and this is the origin, which one is proximal and which one is distal? The origin would be proximal, the insertion would be distal. So origins are usually proximal to insertions. So let's look at those attachments, what attaches a muscle to a bone. We have 2 major types of attachment. One is called a direct or fleshy attachment. It's when the epimysium fuses right to the periosteum of the bone or the perichondrium of the cartilage. Here's a good example. Look at the temporalis muscle. The superior edge, you'll notice there's no white thickening or no tendon there, and that's because the epimysium doesn't come together as a strong cord, but actually adheres right to the bone's periosteum. The more common type of an attachment is an indirect attachment, and that would be like at the bottom of that temporalis muscle where all those membranes merge together and make a rope-like tendon. Sometimes where all the muscles merge, it's not a rope-like attachment, but more like a sheet of connective tissues, and that would be called an aponeurosis. It attaches to bone, cartilage, or the fascia of other muscles. The more common is, of course, the tendon. So now that we've looked at kind of the macroscopic anatomy of this skeletal muscle, let's go and look at the microscopic anatomy. Remember that a muscle cell is also called a muscle fiber. So what is a muscle fiber? A long cylindrical cell with many oval nuclei and a plasma membrane or cell membrane called the sarcolemma. Remember that prefix sarco has to do with muscle. So we're just giving the plasma membrane of the muscle fiber a special name. Now, muscle cells are extremely large cells compared to other cells in your body. They're about 10 to 100 micrometers in diameter and can be up to 30 centimeters long. That's about a foot long. So when I say those little strings are muscle fibers, I really mean that they can be that long, and so they can be somewhat visible with the naked eye. So how do we get these extremely large cells? Through a process called syncytium. Syncytium. Let it say it for you.
Syncytium.
Syncytium. Syncytium is the fusion of hundreds of embryonic cells. You have what's called a myoblast in the embryonic state, and a bunch of myoblasts will actually hook together to become this very large skeletal muscle cell. The old thought process is that they went through several mitotic divisions without cytokinesis, but now the more modern view is that these embryonic cells actually fuse together. So you have a great big long cylindrical cell with lots of peripheral nuclei. Just like we gave the plasma membrane a special name, sarcolemma, we're going to give the cytoplasm a special name as well, sarcoplasm. It contains many glycogen stores. That's where the glucose is going to be stored as glycogen. Those are glycosomes. That's going to give us something to break down into ATP. And aerobically, we're going to need oxygen. So just like a red blood cell has hemoglobin that holds on to oxygen, muscle cells have myoglobin that holds on to oxygen. Each muscle fiber has many rod-like what's. We've already talked about this. Muscle fiber is made of myofibrils, and they run parallel to each other down the entire length of the cell. So what is a myofibril? There are many myofibrils in the cell, and so many they actually push the mitochondria away and push the nuclei to the outer edges. That's why they're peripheral. If you look at this picture, you can see how the mitochondria are all squished in between these myofibrils. Now, the endoplasmic reticulum is actually going to run around each one of these myofibrils in a very elaborate form. The ER, or endoplasmic reticulum, of a muscle cell is called the sarcoplasmic reticulum, and you can see they've colored it blue, how it runs around each one of those myofibrils like a sleeve. Now notice that going down through the sarcoplasmic reticulum is a white structure that they've colored white. It's called the T-tubule, and it's an invagination of the sarcolemma. So it's the membrane on the outside, and it goes down deep inside of the cell. And the reason for that is so that when a nerve impulse or an action potential is sent, that action potential will not just stimulate the muscle fibers or muscle cells or myosin and actin on the periphery, but will travel deep inside of the skeletal muscle cell, and they'll all contract together. So that T-tubule. On either side of the T-tubule, you'll notice that the endoplasmic reticulum is enlarged. Those are called terminal cisterns or lateral cisternae, and the job of this ER that's so vital to the functioning of the muscle is to store and regulate calcium, because two things you need for a muscle contraction is calcium and ATP, and we'll see that later. So here's just an enlarged view. You can see that the T-tubule and the two lateral cisterns are known as a triad. And so that's how we're going to hold on to calcium. So when that nerve impulse comes, it's going to go down the T-tubule, and it's going to excite all of the sarcoplasmic reticulum to release its calcium. So let's look at the notes that say just what I just got through explaining. The sarcolemma invaginates and transects the myofibril. These are known as T-tubules. On either side of the T-tubule, the sarcoplasmic is enlarged and called terminal cisternae or cisterns. The T-tubule and the two lateral cisterns make up a triad. The T-tubule functions to transmit the nerve impulse deep into the fiber for a more rapid response. So we have the muscle, the fascicles, the fibers, the myofibrils. So what are myofibrils made out of? Alternating rows of myofilaments. Myofilaments are rows of proteins arranged in alternating rows where the ends overlap to give the muscle a striated or stripe appearance. So looking at our picture at the bottom of the page, you can see there's the thin actin and then the thick myosin, the thin actin and thick myosin, and thin actin and thick myosin, and thin actin. And so they're overlapping each other. They're alternating rows that overlap. The thin one we know is actin. It's about 8 nanometers in diameter. And because when you're looking at only where there's actin in this muscle, more light can pass through it because it is thinner. So if there's more light, it's going to have a light appearance if more light can pass through. But if you look over here where the thick myosin is, it's 16 nanometers in diameter. Less light would pass through the myosin, so it would have a dark appearance. And so that light and dark gives you the striations or the stripes. Now looking at this functional unit of the muscle, you can see a third type of myofilament. It's what they have yellow. It looks like little springs. That's called titin or elastin. And what it does, titin and elastin, is that it actually stabilizes the myosin and gives you elasticity. So this is the actin, this is the myosin, and this is the titin or elastin. So looking at this unit, this is actually called a sarcomere unit, and it is the basic functional unit of the muscle. If you get any smaller than this unit, you can't see how the muscle functions. You just have individual proteins, actin and myosin, but this is as small as you can get and still see those pieces working together. So let's name the parts of a sarcomere. This area right through here, if you were to shine a light down from the top, you would have more light coming through this area because you have relatively thin myofilaments in this area. What is the vowel in light? It is I, so they named this portion the I band. This portion of the sarcomere unit, if you shined a light down through it, not as much light is going to come through it, so it's going to appear dark. What is the vowel in dark? A, so that's the A band. The area where two actins on alternating rows attach is called a Z line. And so technically from one Z line to the next Z line is a sarcomere unit. The area where the two myosin groups join in the middle is called the M line. And the area of the sarcomere where there is only myosin when you look down is called the H zone. So imagine if the overlapping increased and we slid together, we would lose our H zone and our actins would move towards the M line. And so the Z lines would come together and our sarcomere or our whole muscle unit would shorten. You will be labeling one of these sarcomere units on your test. So let's put it in note form, what we've just described in picture. A sarcomere is the functional unit of muscle. It extends from the middle of one actin or light band to the middle of the next actin or light band. This middle is known as the Z line or the Z disc. The light area is called the what because of the vowel? The I band, while the darker is called what? The A. In the middle of the dark band or the A, where there is only myosin, is the H zone, and the center of the H zone is the M line. From one Z line to the next Z line is considered a sarcomere, the functional unit of a muscle. So let's look at each part in a little more detail. Let's look at the myosin first, the thick one. Notice that each myosin myofilament has two heads whose tails are interwoven. It's like we take two golf clubs and we spiral the two ends of the golf club that you hold on to, spiral them around. Now the tails of the myosin are actually pointing towards the M line. Let me just go back to this picture. Do you see here how the heads are here? So the tails are pointing towards the M line. The heads are known as cross bridges because they're going to actually attach to the actin and cross the space between the actin and the myosin. So they're known as cross bridges. The head contains three special features. On the head, you're going to see the part that attaches to the actin. It's called the actin binding site. Then you'll see an ATP binding site because in order for the myosin to actually be cocked into position and then have a power stroke, we have to have energy. And then we also have a very important enzyme in that head called ATPase. ATPase is the enzyme to break ATP down and to release the energy needed for the contraction. So let's look at the actin molecule. The actin molecule kind of looks like a string of pearls, two strings of pearls that are, twisted around. And then running through the string of pearls, there's two gold chains. And on the gold chains, every once in a while, is a gold bead. Well, the pearls are kidney bean shaped monomers that are held together in a long double coiled polymer. That is the actual actin filament, like a twisted double strand of pearls. And then the gold strands that are woven into our necklace, so to speak, those two golden strands are called tropomyosin. And every once in a while along the tropomyosin you have a troponin group. The tropomyosin group, the gold chain, actually covers up the myosin binding site on the actin, and the troponin, which is like the gold bead, is a 3-polypeptide complex that will bind to calcium, and when it does, it will pull the tropomyosin down so the binding sites are exposed. So when calcium is present, the tropomyosin group is pulled down because the calcium attaches to the tropin, and now the myosin binding site is exposed, and if that myosin is there and cocked because of ATP, they will attach to each other and a power stroke will happen, and their overlapping will increase because they will slide across each other. So let's look at titin, or that elastic filament. It extends from Z line to the thick filament and then to the M line so that it can hold the myosin in place. It can help with elasticity, so once the contraction's over, we can go back to our original length, and it resists excessive stretching so we don't become too limp and unable to contract. Actually, if you've read your book, you know that there are other myofilaments as well. They're just not as important as the three I've already mentioned. They help to hold the sarcomere together. I didn't want you to think that I didn't know that there were more myofilaments, but they're just not as important in understanding the contraction. So I like to show this picture because this is looking down into the end of a myofilament or a myofibril. Not myofilament, myofibril, and you can see how the filaments are arranged in a very pretty geometrical pattern, where the thick one is the myosin and all the way around it are actin. So if you can imagine that all around the myosin you have those heads that will reach out and cross the space to the actin and bridge them together and slide the actin across the myosin. It would be almost like if you had a pipe over a pipe and you slid one through the other one. Very special geometric shape. So we're going to pause right here before we go into the physiology or the contraction of the skeletal muscle.